everybody. I'm Samuele Marro from Mount Sinai. Again, in our lab, as you know already, we study DNA and what happens when you change DNA and when you manipulate DNA. So to go back to our previous lesson where we learned about, you have seen this slide hopefully already, we learned about the DNA being a very complex object, but yet very simple because it's only made by 4 letters, TAGC. So I want to start from there and go a little bit ahead from what we said. What is it needed for? DNA has the instruction to produce proteins. If you just sequence the DNA, and we do this in the lab every day, you read this almost infinite number of repeated sequence. It's really hard to wrap your brain around it, to make sense of it. Look at your screen and tell me if you can catch anything that makes sense to you. It's really hard to find patterns. Of course, you can find the repetition of 3T, but then, next one is CAG here, and here is GGT. So this is part of our job, right? Trying to make sense of it. And of course, we use computer and tools to help us, all right? So this is the concept that I want you to understand, that the DNA is very simple, but yet very complex. If we understand the code, we can actually make sense and understand the information is in there. And this is what we've been doing in the past 40 years. So let's try to understand a little bit more how this code works. I told you, it's the instruction to produce proteins. And usually, when you talk about proteins, bunch of meat, eggs, if you look at this picture, what do all these things have in common? These are all living things. Living.
Well, once living.
Right? Once living, unfortunately. So what do have living things in common? They have cells. Cells are made of proteins. Yes, they have other things, they have fat, they have DNA, but really the key components of the cells are the proteins. And to give you a sense, I want to play you this video, which I think is very cool. Okay, it's a modeling of real proteins inside the cells. And specifically, it's 2 types of proteins. One is called kinesin, comes from the Greek term, means moving. And the other is the protein called microtubule, which creates this very long thing, creates the structure of your cells. All right.
So the long thing is the microtubule, and is the blue thing?
That is actually a vesicle. And a vesicle is like a bag that the cells use to communicate between cells also, right? When right now we are speaking, we are thinking, it means our neurons are exchanging exactly this, exchanging these vesicles, right? They are giving vesicle to another one, like, hey, this is my message. There are little information, very interesting biology behind vesicles. You will see that this protein, this microtubular protein, interacts, it means is attached to the kinesin protein to do a job. And so the result is actually carrying around this vesicle that is needed somewhere, right? So let's play the video and look at how the kinesin moves, literally carrying the vesicle.
It looks like an elephant's trunk.
It looks like an elephant. I think it's very, very cool how it works. But this is not a cartoon. This is in real life how our proteins work inside the cells.
Wow.
So this is obviously probably a lot more advanced than most of the, microscopes they use in school. Yeah. And what will eventually happen in a couple of minutes, seconds hopefully, is that we will have to initialize, this is what we call the stage. So this joystick actually just moves around the stage. And basically what will happen is that we'll get a slide and I'll put it in there and it'll stay in. And then depending on which, where the brain is located on the slide, I can just move the stage. So I'm gonna hit live. We're now gonna reset the focus because now we have, jump magnifications. At first we were at what we call a 5x magnification. So we were super zoomed out and now we are zoomed in. But kind of in the same way that, when you're too close to something, like, it kind of just looks like it's in your face, right? So we can't really see it that well. Cool. And don't worry, it looks blurry now compared to what you're looking at on the eyepiece, but it's always, the focus is always a little bit different between the digitized camera and what your eyes can see on the eyepiece. So
what is kind of your guys' goal with the research that you're doing here in the lab?
Our lab in particular, we study neurodevelopment. and we study specific genes that are mutated or, seen to be mutated, in autism and Down syndrome. And so we study how having too much or too little of these genes can affect normal development. For example, you might be familiar with Down syndrome, and how there is an extra copy of chromosome 21. So one of the genes that our lab studies is part of the critical region in that chromosome 21 that Down syndrome patients have an extra copy of. One of the overarching goals of our lab is to understand what is this gene? How is it important for neurodevelopment? What happens when, we take it away or add too much of it, or when we mutate it?
In terms of your team here, obviously, who do you have here? Who do you work with, or do you mostly do your work alone when you're here in the lab?
So science is a pretty collaborative environment. Me, myself, I work across multiple different projects. We have a mix of we have a couple different grad students, PhD students, and some master students. So whenever they need help, I'll help with their projects here and there. I also have some projects of my own that I kind of spearhead and take charge of. But most everyone in the lab kind of, at some point or another will help each other out with their own experiments.